examples of our scientific curiosity. So these are the lineage-specific differentiation which I was mentioning yesterday, and the kind of cell type that we obtain. What are the other reasons that one rather does with stem cells in terms of research and their potential application? This gives an overview. One, of course, is the differentiation biology to achieve neurons, to achieve some blood cells from mesodermal lineage, to achieve cardiomyocytes, to achieve pancreatic beta cells, and so on and so forth. It is also important that these cells can be used as an in vitro model to differentiate the cell types and then see what kind of drugs will benefit differentiation or inhibit differentiation. Both are required if you are doing drug development, depending on the disease condition. So it's a very excellent experimental-based model system to look at a number of approaches. The pluripotent stem cells, whether it is ESL or iPS, is also used for a number of toxicology studies. Traditionally, even currently, the toxicological research, whether there is anything harmful of a drug or anything, is done in small laboratory animals. So one can minimize the use of small laboratory animals by using an in vitro culture systems. In other words, you use the pluripotent stem cell, differentiate them to a particular tissue or organ if possible, and use this as a organs in petri dish, organs developed in vitro, which can now be exploited to test a number of toxicological compounds for safety or efficacy or what have you. So that's the whole thing. So one reason, of course, is to produce these different cell types. The other reason I will tell you is this is just to depict you how one can overcome, let's say, a hematological disorder, sickle cell anemia. Somebody is suffering from sickle cell anemia. I'll give you one classic paper that I published some time back, but this will give you an idea. Let's say you have an experimental system. You can consider here the mouse. You can also think that this particular cartoon can be human. So you have a sickle cell affected individual, in this case, let's say a mouse. So how do you treat a genetic disorder such as sickle cell anemia or hemophilia or any other hematological disorders which are genetic at the cause, or a particular gene or a monogenic or a multigenic cause or causes for that particular genetic disorder? Now, the way to do that is to take the fibroblasts, like I said, you produce induced pluripotent stem cell. So that induced pluripotent stem cell will have intrinsically that particular gene defect. Now, these cells can perpetually be grown in the petri dishes. So now what you do is take some scissors, molecular biology scissors, cut the defective gene. And in that little gap, insert the correct gene, which you know, molecular biologists and genetic engineers know exactly the gene segment. Now put the correct gene, which is functional, and put back and stitch it. So I said cut, introduce the corrected segment gene, and stitch it. These are the three approaches. It's exactly like a tailoring job by a molecular biologist. And therefore, you can correct that defective gene and then get the normal gene in the ES cell population. And there are a number of methods. One of the latest approaches is CRISPR-Cas9 technology, which is an extremely powerful technology. And the two ladies, I forget the names right now, I'm sorry about it, who got also the Nobel Prize a couple of years ago. Now, by CRISPR-Cas9 technology, you can remove the defective gene, you can put the corrected gene, and therefore you have iPS cells which are genetically normal, rather genetically engineered to become normal. Now, these iPS cells can be differentiated to hematopoietic lineage, and therefore from hematopoietic lineage it can be differentiated to erythroid lineage, that is, which can produce red blood cells. And therefore, when you have the erythroid lineage cells transplanted back into the recipient or the defective hemophilic or sickle cell anemic person or animal, then the sickle cell anemia phenotype is reversed. So that's the concept of, or rather potential of stem cells, which can be derived from genetically defective individual, correct that gene, put the right gene, and make those cells to become the cell type of your interest and transplant back into the same individual or patient, and that disease phenotype is reversed. I am not giving here a scientific fantasy by this particular cartoon. It is indeed doable, and it is done in the mouse, and it is being done in the human. All right. There are a number of other potentials of producing cell type specific or other tissue or organ type specific cells and tissues, and so on and so forth. We in our lab have done quite a bit on cardiac myocyte differentiation, and this was almost about 12, 13 years ago or 15 years ago. I don't remember. So we were the first to derive EGFP expressing ESLs and IPS cells, and then the country required national guidelines how human stem cells can be studied, researched, and potentially exploited to do clinical trials, so on and so forth. The national guidelines had to be developed. So the entire country, we traveled and we popularized this, the need for national guidelines, so on and so forth. That's a different story. At that time, the news media people were there to figure out what's going on, and NDTV fellow Bagla was there in one of those meetings, and then he got interested in the type of research. So what I'll show is his own NDTV coverage clipping of what we did, and the reason why I'm showing is that 15 years ago, he made few statements, he forecasted. And when you look at that forecasted statements currently, it makes sense. And that's the reason why I'm just showing this here. I hope you can listen it.
Sir?
Yeah.
So we couldn't hear.
You can't hear?
Yes, sir. We couldn't hear, sir.
I don't know, but I will just run through. I can't do anything now. I'll just run it through though. You can, I probably will explain my own terminologies. I have shown these yesterday actually. This is skeletal muscle. This is almost 15 years ago. So all this work and coverage was on our EGFP expressing transgenic mouse, and from the transgenic mice, our derivation of embryonic stem cells, our derivation of iPSC cells, and demonstrating that the heart muscles cells can be produced, nerve cells can be produced, and he explains that we cannot get the tissue and organ yet. He said there's a huge potential. He says human stem cells can potentially be now extrapolated to produce organ-specific tissues or even organs. But that time, producing organs is too far-fetched. You know, that was almost 15 years back. I should thank NDTV and Palla Bagla and his team to cover this almost 1.5 decades ago. So I just listing here what is the great potential of stem cell based potential therapeutic options that can come, and I have listed a number of diseases. You name disease, there are potential possibilities of using stem cell derived cell types for alleviating the disease phenotype, whether it is cardiovascular disorders or diabetes mellitus, Parkinson's disease, or Alzheimer's neurodegenerative disorders, ischemic condition, blindness, stroke, spinal cord injury, to have supporting as well as neuronal cell transplantation, critical liver diseases, chronic liver diseases, chronic kidney diseases, musculoskeletal disorders, autoimmune disorders, hematological disorders. Probably I'll give you 1 or 2 examples of these. So the potential is really very huge. And yesterday I showed this slide, and the basic academic research. One of my students, when he was trying to look at the fluorescence of these cells, he used obviously monochromatic light. And when he used it, and he realized that if he continuously looks at the monochromatic light to look at the fluorescence, the heart cells, the cardiomyocytes, ceases to beat. So obviously it was a fantastic discovery that he made several years ago, that a monochromatic light, if it is continuously exposed to cardiomyocytes, ceases to beat. But when you expose them back to a normal light and put back the petri dish into the incubator, it recovers back. So it's a transient cessation of beating property, which is recoverable if they are not. So it takes some time. So the entire time dynamics and biochemical basis for this have all been published. And this was a fantastic curiosity-driven research. That's the reason why I'm giving that example. And that got published a couple of years ago, if I recollect. It was a fantastic study, but there may be huge implications. And we were able to demonstrate in the human cardiomyocytes derived from iPS cells. This may have some clinical implications. I will not go into detail. I have just listed them there. But these are some kind of a scientific curiosity-driven research outcomes which may have a great clinical potential. And one of the examples I mentioned yesterday is about protection of cardiomyocytes, both in the 2D cultures, and I said that they just float. These are completely differentiated cardiomyocytes from human iPSC cells. And we also cultured them in the PCL scaffold. And I said this can be stitched in ischemic heart diseases. And using that, we need to demonstrate that this is actually experimentally can be proven. And therefore, my collaborators and my own student who is there in Ohio State University and working in Dr. Mahmood Khan's lab have been able to do this in the human iPSC-derived cardiomyocytes or cardiac progenitors, and then see that the ischemic condition of the heart in the rat, for example, or it could be mouse or rat, it can be alleviated. And very recently, a couple of months ago, I was there, and we were able to demonstrate this even in the large animals, such as the pig. So, in other words, what I'm trying to tell is this iPS cells can be differentiated to become cardiac progenitors, cardiomyocytes. This can be transplanted to experimentally induce ischemic heart containing large mammals as well as the small mammals, and there is alleviation of cardiac ischemic conditions. So what it tells is, if you have that little cardiac patch, which is PCL based, and if it is human cells, obviously in this case it is human cells, and if these are clinical grade human cells, if they are ready to be transplanted after evaluating its safety and efficacy becomes known by these animal model experimentation, then it is possible that a clinical grade cardiomyocytes, which are cultured on the PCL patches, can be transplanted into ischemic heart condition, which is myocardial infarct condition. Therefore, the potential of a heart attack can be completely eliminated or minimized in the individuals who are suffering from ischemic heart diseases. And that's the potential of using either the 2D or the 3D culture system to get the iPSC-derived cardiomyocytes for potential transplantation into the human. Now, if you look at that kind of a thing, the number of diseases, like I mentioned, there's so many diseases which I listed earlier. And I'll show you one picture. I missed it. I missed it somewhere. Okay, that's fine. Okay, so that's one typical example. And this is the hemophilia. I showed you one cartoon picture where you can take a somatic cell from the sickle cell anemic animal, do cell engineering and genetic engineering, and have a genetically normal cells transplant and reverse back. That is indeed was demonstrated in rodent models, and these are the papers that came out. These are very classic papers to show. So which means the hemophilia or sickle cell anemia, the patients who are suffering from this can have a remedy by this so-called a combination of a cell-based and gene therapy-based approaches for treating genetic disorders. And in fact, Christian Medical College in Vellore have been very instrumental in producing clinical grade iPS cells from the patient and try to do genetic defect corrections and then do transplantation in hemophilic patients or so on and so forth. So the opportunities of potential application of iPSC-derived cell types, both for cell deficient problems as well as gene deficient problem, can be overcome by this combination of stem cell based as well as gene targeting based approaches for even genetic disorders. A similar approach has been used for HIV patients alleviation. I will not go into the detail. It's a little complicated. You need some more time if I need to explain. Basically, it's possible to dupe the immune cells from the HIV positive patient, do a genetic correction to that, and then put back, and therefore that person will have minimal HIV infected phenotype or eliminate that one. So I will not go into that. That can be corrected. So by doing this mechanism, you will actually kill the HIV virus or RNA virus. So there is an approach to it, and I will not go into that detail here. The other method, an example that I'm going to be talking about is, let's say there's a type 1 diabetes. And whether you, in type 1 diabetes, the individual, it's also true in humans, for some reason, the kid is born with pancreas, of course, but with a pancreas which either doesn't have sufficient numbers of islet cells, beta islet cells, or have islet cells in minimal numbers but do not produce insulin. So they become completely lifelong insulin dependent exogenously. So those individuals have to be given insulin every day, twice or thrice in a day. And imagine the plight of the quality of life of such an individual. But there are people, there are a number of lakhs and millions of people who are affected with type 1 DM, and they do live normal life thanks to insulin being injected. But that cannot go for lifelong, for 60 years, 70 years. There are other complications. I will not go into detail. But is it possible to get the beta islet cells produced, or even that pancreatic organ produced, and therefore do islet transplantation, or pancreas which contains normal number of islets, beta islets in that organ? So transplant the entire pancreas. To do this in the human, you need to experimentally demonstrate in vitro condition, in vivo conditions, in model organisms, so on and so forth. Here is a beautiful example where an interspecies organogenesis for xenotransplantation can potentially be carried out. I will not go into great detail. So it's basically to show here that one can make an animal, let's say a rat, completely apancreatic, if during development, you delete the PDX1 gene. PDX1 is a transcription factor which determines the endodermally derived pancreatic cell type. And if that gene is not there, you will not get pancreatic tissue and obviously will not have beta cells. And therefore, when you take that kind of a thing and transplant those blastocysts to a recipient, you will generate rats or mice which will not have pancreas. That's a control. So if you take those blastocysts and put another animal species normal gene, PDX gene of the mouse into the rat iPS cells and regenerate blastocysts, which is a chimeric blastocyst. I explained yesterday about chimeric blastocysts, and transfer that chimeric blastocyst to a recipient animal. The offspring born out of that will have a rat pancreas produced by the human, produced by the mouse. Let me put it the other way. You have the you will be able to produce a rat capable of producing insulin out of this hybrid approach. And so this is possible because you are able to make an organism apancreatic, like some individuals who cannot have pancreatic beta cells. You can take out the somatic cells, fibroblasts, engineer those fibroblasts to become iPS cells. Those iPS cells can now be introduced with the normal PDX1 gene so that you will have a lot of iPS, a lot of beta cells to be produced, and then put that into, let's say, an animal. In this case, it can be pig. And the pig will produce the human pancreatic tissue. And then you use the pig as a donor to take the pancreas and transplant the pancreas into the human. So these again are not fantasies, but these interspecies organogenesis has been created very successfully in the mouse and the rat using pluripotent stem cells and this kind of genetic engineering approaches. And these are actually possible, done even for the human, using the human iPSC cells, and therefore human pancreas is produced in the porcine system. That this particular porcine individual or a species can become an organ donor for transplantation into the human. So these are very remarkable discoveries that have been made by stem cell researchers and stem cell technologies. So basically what I'm trying to consolidate here, by an interspecies approach, you can produce human organs in a xeno system. Let's say a pig as a surrogate. And therefore, it is possible to exploit the use of iPSC cells, genetically engineer the iPSC cells to make sure that those iPSC cells can in fact produce pancreatic cells, exocrine pancreatic cells, endocrine pancreatic beta cells, which are capable of producing. And the organ per se can be formed only in an organism during development, during fetal development. And that's why you use this approach. But whether you can produce an organ in vitro without the involvement of an animal development, yes, that's possible by what is known as 3D organ printing. And I will not go into that detail, but there is a way to do. You need to make sure that all the cell types of the organ, which are critically required to form the tissue and the organ, would need to be individually be pumped into that printer. Like you have an inkjet color printer having 4 different colors. Similarly, you will have 4 different cells, and they put it in some proportion, and therefore they will layer after layer at level in a particular scaffold. So you can generate organs in an in vitro condition by 3D printing mechanism. That's where bioengineers, mechanical engineers, and medical biotechnologies come together to do this kind of approach. So these are very important things. And one can ask a question, why can't we simply take an organ and then transplant? Why should you use stem cells and go through the whole cumbersome process of generating tissues and organs? Now, there aren't many organ donors. The demand for organs are very huge, particularly for liver, particularly for heart, more importantly, rather extensively for the kidney, renal failure, and therefore renal transplant. Renal transplant are the largest demand, let's say, followed by liver and heart. And therefore, there are very sparse donors that are available. There may be donors due to altruistic reason, humane reasons may give a part of one of the kidneys that they can donate. The other kidney, they may have it for themselves. Similarly, a part of the liver lobe can be donated. Otherwise, those young individuals, unfortunately, who suffer from mortality due to accidents or some other reasons, they can become organ donors. And such organ donors can be used, but the number available is far, 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 far less than the numbers that are required. And so there's a big mismatch of supply versus demand. And that is one big issue. The other big issue is even if there is a donor, that has to have an immune matching. Haplotype matching should happen. But one can do haplotype matching through iPS technology. So I may have one particular haplotype, somebody else may have a different haplotype. I can take my iPSC cells from the fibroblast, different reprogram, and I can change the haplotype. There is a way to do that one. And that is how xenotransplantation technology, let's say the pigs for xenotransplantation. See, the pigs will have their own genes. So what the scientists are trying to do is remove those histo-incompatible genes of the pig from their genotype or from their genome and put histocompatible genes of the human into that genome. And therefore, the pig is produced where the pig haplotype genes are eliminated, human haplotype genes which are compatible for transplant to the recipient is introduced, and therefore an organ developed in that particular organism, in this case, let's say a porcine system, will have an organ which will not have rejecting gene and gene products of the pig, but it will also have accepting gene or gene products of the human haplotypes. And therefore, if that organ is transplanted, the possibility of that being rejected and the possibility of long-time survival of that organ and therefore survival of the individual is very, very high. And that's a big approach. And in fact, that was shown about a year back in the human. So they had a xenotransplantation carried out in the Baltimore in John Hopkins University, where a genetically engineered pig which had haplotype matched genes and which eliminated the haplotype mismatched porcine genes. They engineered that and they made a heart of that in the pig, and that organ heart was transplanted to this particular patient, Bennett, and that person survived, I think, for several months. But 15, 20 years ago, or maybe 20 years ago, baboon heart was transplanted into a small kid, I think. I don't remember the age, maybe 4 years, 6 years. That individual survived. And this was the first example of an adult of 55 or 57 years old who survived post-transplantation of xenogenically produced heart. Well, the last part I just want to cover is how cell therapy or stem cell industries actually are into this R&D and kind of thing. Obviously, there's a lot of money involved. The turnover is very high. The demand of cells and tissues and organs are very, very high. And you can see the trajectory. This is, of course, about almost 8-9 years old, but that the demand is actually huge. The market in terms of $1 million is really huge. And that's true not only in, globally speaking, even Indian market for stem cell based potential therapeutic approaches are very, very huge, because there are a number of patients who really require, and these have to go through clinical trials, and there are methods of doing this. Like I said, safety and efficacy of cells and tissues are utmost important. Otherwise, the regulatory authorities of the government, whether it is Indian government or any country that we're dealing with, US FDA, for example, in the USA, they will not give approval. And without approvals, you should not, and one should not do any clinical trial, any touching of humans with the human cell. It has to go through a lot of ethical regulatory measures. Only then you can do. So this is extremely important. So if one asks, okay, you're still in research and how long it takes, and this is the kind of a time trajectory, it takes anywhere between 12 to 15 years from the bench side, basic research side, to a bedside. For that matter, whether it's a pharmaceutical drug or a small molecule drug, or biopharma companies having biologicals, whether it's antibody or cell or whatever, they take really 10 to 15 years for it to come to reality in terms of getting used in the human. So it's a huge research, huge investment, huge level of expertise, and one needs to follow absolute, rather highest level of scientific, technical, and ethical norms, and comply with the regulatory measures, or in other words, a law of the land would need to be followed if anything is to be done here. Anything that is not followed is completely illegal, not even illegitimate, illegal, and that should be stopped anytime. So, in a nutshell, what I would say, in stem cell research and stem cell potential therapeutic applications, there are a number of people who are interested, and I told you that. So the stakeholders are actually too many in this, and the science is actually too complex. The scientists are trying to solve the puzzle in many, many ways, but it is actually a very potent biological research, very prolific research, and very prospective research, and it has to be absolutely a multidisciplinary stem cell biology research. And so therefore, there needs to be a kind of a regulatory measures to control this. Otherwise, there may be illegitimate quackery that may land up, particularly true in case of stem cells. There are many companies, known or unknown, who claim that they have a stem cell product ready to be given therapy, and there is no therapy so far except for one or two. For hematological disorders, stem cell based therapies are available, but for no other clinical indication, therapy word is not there as far as the stem cell is concerned. Everything is experimental. Everything has to undergo clinical trial. You are trying it. You may succeed and you may not succeed. If the patient is willing to take that testing, he or she can take it up. So it is under clinical trial. When a clinical trial is carried out, that clinical material which is injected into the human should be safe and efficacious. So it has to go through a clinical grade process, and it should be a clinical grade material which are injected. So that is a different ball game. I will not go into the detail, and these regulatory measures are important. So in a nutshell, if you look at it, there are still, from the application point of view, there are still a number of problems or issues that are still to be solved. One is, getting them to isolate the right type of cell types. I said differentiated cell types, their enrichment process, the purity process, which I mentioned yesterday, who will do and what it takes to do, who are the experts who can do, both from the scientific point of view, infrastructure point of view, technical point of view, upstream scaling, and scaling up of producing more number of cells, an infrastructure which is of a GLP or a GMP or GTP types. These require huge investment. So who are going to invest? It will be the government of India, or whether it will be private entrepreneurs, or it's a teamwork of private entrepreneurs and the Indian government. So it's a public plus and private sectors coming together to make a cell-based approach as an alternative or an important therapeutic option potentially for alleviating human diseases. So these are some use. There needs to be a real integrative approach of academicians, clinicians, and cell therapy industries, both the governmental sector and the private sector, to come together to see that this collaborativity can go forward to see the benefits or reap the benefits of stem cells for treating human disorders and diseases. I will not go. These are some of the regulatory bodies that we all should be aware of. At the institute level, you need to have institutional ethics committee, and if you are dealing anything that you want to do with the human, it needs to go through institutional ethics committee. Anything to do experimentally with stem cells, you need to go with the institutional committee for stem cell research. Anything to do with the animals that you are doing, you need to have institutional animal ethics committee. And all these get supervised at a higher level by the government of India. There are apex committees. There's an apex committee for animal research, small and large, particularly large animal research, CPCSCA. And anything that goes into human, the DCGI is the nodal authority to provide the approvals and the license. So I am going to stop here, and I thank the Science Academies and the organizers, Sri Shankara College, for organizing this, and thank you very much. I'm sure.
Thank you very much, sir. The session is now open for interaction. Any doubts? Can you please come forward?
Can we use the stem cells from dead animals? You mean dead animals?
Yes, sir.
Okay, it depends on when, how many hours post death or mortality. So it depends on what cell types you're talking about. Some are relatively long-lived in terms of few hours. Some are very short-lived. So it depends on what is the sourcing of the tissue or the organ that you're talking about, and how sterile it can be to take the, I hope you understand the word sterility. Completely microbial way of, microbial free way of approaching, even if you have a carcass. So all these will determine the viability of cells that are taking, tissues that you're taking. If it is an hour, a couple of hours, it should be okay. But if we are talking about an intestinal organ, let's say you want to take a little bit of liver or a pancreas, the bacterial load will become more there. For example, if you want to take a tissue from the brain, it's much safer even after a few hours. But if you take some tissue from pelvic region or abdominal region, the possibility of contamination is very, very high, microbial contamination. So, and what sterile technique you use to take it out? If it is a dead individual or an animal, we tend to be not sterile. You use any scissors, forceps, scalpel, anything. No. So it all depends on how you approach and reach the organ of tissue.
Actually, all about cancer germs. The cancer germs have a very few cancers. Can they have a very few cancers? We use the radiotherapy chemotherapy only. Nowadays, we also use the cancer. So the cancer stem cell has to give the cancer.
People don't use it, but there is an approach called T-cell based approach, CAR T-cell therapy. I don't know whether you have heard. If you Google, you will know CAR T, C-A-R-T therapy. So that is called chimeric antibody, chimeric antigen antibody. That is basically you're taking the T cells of the patient and engineer the T cell in a manner it will have a cancer killing property, A, and B, it will specifically get targeted to the cancer cell in the body. So you have these CAR T cells, which are actually produced from the T cells from the patient, engineer it and put it back to the patient. What it will do, it will go to the particular cancer site and kill only the cancer cells. But cancer stem cells per se, unless you make it to be less proliferative, non-cancerous, again by genetic engineering approach, and then introduce cancer killing property, then what happens is when you do that, in vitro itself, these cells will get killed. So it all depends on the experimental strategy, but cancer stem cell as a cell-based approach to treat cancer is a roundabout approach. You can as well use T cells of the patient and do genetic engineering.
Sir, my question is, if a patient is actually about to have a transplant, can he use the same person's stem cells to actually produce an organ and fully function one?
Because there's an echo in your auditorium, I'm not able to get clarity. Can you said can you use stem cells for organ? Something you said. Can somebody repeat or slowly talk?
Sir, the question is, if a person is a transplant, can he use the same person's stem cells to produce a fully functional organ for the transplant?
Oh yeah, 100%. I think that's what I was explaining. From the patient, you can take fibroblasts, skin fibroblasts. You just take a little biopsy, do trypsinization, you will get fibroblasts, somatic cell. Then you introduce some genes, and that will produce iPS cells, that is stem cells, of the patient. And then you make that iPS cells to form whatever cell type that patient requires in vitro, and then put those cells back into the same patient. Like I gave in my initial talk, if I am diabetic and I want my beta cells, I will take my skin fibroblasts and I will get my iPSC cells, and I will get my iPSC derived islet cells, that's beta pancreatic cells, and I will transplant it to my body inside the pancreas, or I'll put it subcutaneously, maybe somewhere here or here. I can do it. So it's patient specific, patient derived, patient requiring cells.
Good morning, sir. Sir, here's my question. To test the toxicity or the efficacy of the drug, just use one sensor to test in order to avoid the animal killing.
You are talking about toxicity, stem cells. Can you talk slowly and let me recapitulate what your question is?
Test the efficacy of the drug or else toxicity of the drug. That means just use only the sensor to test in order to avoid the killing of animals.
Oh yeah, I mentioned that in one of the slides. I did mention about it, that toxicological studies can be carried out on stem cells. Earlier, historically, it used to be done on animals like rats and mice. That can be minimized, because if a particular compound your laboratory has produced, which is liver toxic, so what you can do is produce liver cells from stem cells in the lab, and test the compound whether it is toxic to liver in the petri dish in your experimental setup. So in that way, you will minimize doing those experiments in rat or mice. So toxicological testing, drug screening can be done using stem cell derived cells in vitro without animals.
Thank you. Good morning, sir. Epigenetic changes, how is the stem cells, sir?
Are there epigenetic changes in stem cells, is what you're saying?
Does the epigenetic changes affect the stem cells, sir?
Okay. The question is, if I am correct, are there epigenetic changes that occur in stem cells? Is that the question?
Affect the stem cells.
Right. Does epigenetic changes affect the stem cells? This possibly is true in iPS cells. There can be epigenetic changes that occur in iPS cells because the genomic stability and erasure of some gene expressions are very somatic cell specific. Because we derived iPS from there, people say that there are some epigenetic changes that may occur in iPS cells, but it is very, very less likely in the ES cells. So epigenetic changes can occur anytime in any cell, depending on the environmental insult that the cell sees. So that could be true for any cell, but it is very less in ES cells, but maybe more in iPS cells. But we need to do the analysis, and if that is really affecting your experiment or outcome.
Thank you so much. Any more questions? If there are no more questions, next I'd like to call upon the data sheet of second MSc to end session 6 with board documents. A very good morning to all. It gives me an immense pleasure to deliver the thanks for this event to all dignitaries here. I would like to thank our Honorable Chief Guest Professor P.D. Seshagiri. Despite his busy schedule, he has found time to raise his information. And I also express my heartfelt thanks for his valuable contribution. Today, your ideas and elegance of explanation on topic stem cell biology and technology and applications, treating of genetic disorders, risk therapy, and cardiogenesis, organogenesis, cell therapy, treatment of sickle cell anemia, mouse model with iPS cells generated from autologous are very useful, and this will enhance our knowledge. Once again, thank you so much, sir, for your inspirational words. Thank you.
Thank you.
Thank you, Ms. Devashi. Now let's refresh ourselves with a short tea break. I request everyone to be present
Bye-bye.
at 45 a.m. for session 7. Thank you.